Chapter 31 is getting back to our systems, going through the nervous system. All right, so our nervous system has two main parts. So it has neurons and then supporting cells. Looking first at the neurons, there are what are called association neurons or also interneurons, and those are present in most invertebrates, but all vertebrates. They're located in the brain and the spinal cord, and the brain and spinal cord collectively make up the central nervous system. And those interneurons especially help with complex reflexes, in the case of the brain, that include learning and memory. So in addition to those interneurons or association neurons, there are two other types. So sensory or afferent neurons, those are the ones that carry impulses from sensory receptors to the CNS. And then we have motor or efferent neurons, and those carry impulses away from the CNS to the effectors like the muscles and the glands. So they're actually producing an effect. So efferent effects is one way to try to keep those together. So you have your interneurons, association neurons, and then you have your sensory neurons that take information to the brain, motor neurons that take it away from the brain. So together, the motor and the sensory make up the peripheral nervous system. So the association neurons are in the central nervous system. The sensory and the motor make up that peripheral nervous system. Just showing these working together. So let's say we have an impulse, so we touch something hot or something pointed. So that signal is then going to be carried up through that afferent neuron. It's going to go up and then release a signal onto this association neuron. That's going to send the signal on to an efferent neuron, and then we're going to carry that signal down the axon, we'll talk about here in a minute, and that'll cause the release of neurotransmitters that cause an effect on something like a muscle or some kind of gland that is going to be happening. So this would be the effect that would be produced. All right, so just kind of the organization of the vertebrate nervous system, which is what we're focusing on. So again, our sensory nervous system, that's the afferent nerves. It is carrying information to the central nervous system, so either the brain or the spinal cord. Then that information is processed, and then a signal is sent through motor neurons or efferent nerves, and that is then going to direct muscles, organs, glands, different receptors to do something. And then those can then send signals back to the brain. So we go through this continuous kind of cycle to allow information to be processed. All right, so a little bit more about our neurons. They have essentially three main parts. They have a cell body, which is going to be where the nucleus is. Then they have dendrites that form highly branched structures, and they have multiple dendrites, and then they have an axon. Most of these get support from what are called neuroglial cells. And a couple examples of these, Schwann cells and oligodendrocytes are a couple examples of these supporting cells, and they are going to produce kind of a fatty material called myelin, and myelin is going to wrap around the axon, and it creates an electrical insulator. So just like a wire, it gets insulated so that it can conduct that electrical charge farther. If it doesn't have any coating on it, it doesn't send it very far. Same thing with our axons. That electrical signal from our nerves, this insulator from the myelin helps with that. So some of these others, so again, the Schwann cells produce myelin. Again, this is peripheral nervous system. Oligodendrocytes do the same thing, produce the myelin, but in the central nervous system. So again, they're doing the same thing. They're making myelin, just in different parts of the nervous system. That myelin is going to wrap around axons, creating what's called a myelin sheath, and it's multiple layers, again, to provide that insulation or that conduction protection. It doesn't need to be continuous all the way down through, or else the signal wouldn't make it from one end to the other. which we'll talk about more in a second. But it gets interrupted in intervals where we have little gaps where it's not myelinated, and those gaps are called nodes of Ranvier. And again, the reason we need this is because that nerve impulse that we'll talk about, called an action potential, it jumps from one node of Ranvier to the next, and that increases the speed of transmission. And then just a couple of kind of diseases related to this: multiple sclerosis and also Tay-Sachs disease. Actually, what happens in both of these is that myelin sheath degenerates. And just like an electrical circuit, if that insulation wears off the wires, it doesn't make the right connection. Same thing in these conditions. Nerve impulses are sporadic or incomplete because they can't make it all the way down the axon. All right, again, just kind of looking at this in a little bit more detail. So again, our cell body is up here. That's where the nucleus is going to be. Multiple dendrites, so it's called a dendritic tree. All of these little branched regions that feed into the cell body. They receive information, and then they send it down a single axon, which is shown here. But around the axon are these layers of myelin that make up this myelin sheath. Again, it's not continuous, though. It forms these different sections. It's myelinated. And then we have these gaps shown here where there is no myelin, so unmyelinated sections, and those are called the nodes of Ranvier. So the electrical signal literally jumps from one to another into these nodes until it gets all the way to the end. So it just hops until it gets to the end, and then we'll see that'll cause neurotransmitter release. So how these nerves actually make an impulse. So again, it's similar to an electrical wire. It's an change in electrical charge. So at rest, there's active transport happening in sodium-potassium pumps. Again, just a quick refresher, active transport means energy is required because we're moving something from a low to high concentration. And what these pumps are doing is that the neurons transport sodium, which is Na, out of the cell, and they take potassium into the cell. So sodium's going out, potassium's coming in. Now, again, these are charged molecules, so these ions cannot pass easily through the membrane because of those hydrophobic fatty acids. So they can't come back in, and that causes a buildup of sodium outside of the cell. The potassium then gets concentrated inside of the cell, but not quite as much because there's actually a little diffusion that can happen with those potassium through some channels. So it's mainly the sodium that really gets concentrated outside of the cell, potassium more inside of the cell. There are some other ions in the cell, and there's also DNA inside the cell that's negatively charged. So ultimately, there's a charge difference between the inside and outside of the cell due to these ions, and that creates what's called the resting membrane potential, just meaning the difference between the inside and outside of the cell. And actually what happens is the outside of the cell is more positive than the inside. So if we think about a cell here, the inside is actually going to be slightly negative compared to the outside. And then the outside, of course, again, is a little bit more positive. So it creates this potential, this difference in electrical charge where it's more positive outside and more negative inside. Now this impulse, they're constantly using that energy through those pumps to maintain that resting membrane potential. So pumping sodium out, potassium in. So if there's a net or overall negative charge of most proteins in the cell, again, adds to that charge difference, especially DNA, because it is really negatively charged. And there's a lot of DNA in the cell. So there's actually a difference of about -70 millivolts. So that's the resting membrane potential before any type of impulse would occur. All right, we also use what are called voltage-gated channels. So remember, ions can't cross the membrane because they're charged. They can't get through those fatty acid tails. So they need a transport protein or a channel to help them with that. Well, these channels don't remain open though, so they have a gate on them that regulates whether any movement can occur. And this is essentially gated or regulated by changes in the voltage. That's why it's called voltage gated channels. Okay, so as ions or the electrical current changes across the neuron, then that can open these voltage gated channels. They're looking for a change in that resting membrane potential. So the impulse actually starts when pressure or some type of sensory input disrupts that membrane potential, makes the inside a little bit more positive than it was, and that's going to cause these voltage gated channels to open up, and the sodium particularly opens and allows sodium to rush in to the neuron. So these sodium channels, whenever they open, sodium floods in because it is going from a high to low concentration. We'd already pumped out the sodium, so now it wants to come in from a high to low concentration, but also it's a positively charged ion or a cation moving into a negative environment. So there's an electrical difference. So that means there's a huge rush, a huge push for sodium to enter. And what happens when sodium rushes in is the membrane becomes depolarized, meaning it's a little bit more positive. Because again, now these positive sodiums are coming in, so now it's not so negative inside there anymore. So we say it's depolarized, it's changed poles. It's now more positive inside, more negative outside. So those open sodium channels in a small patch of membrane remain open just for a little bit. It's only half of a millisecond. It's so quick. So if that voltage changes is great enough, then that opens nearby voltage gated sodium and potassium channels, and it triggers this kind of domino effect. And ultimately what happens is that we get the generation of what's called an action potential. So whenever the sodium channels first open, we see this wave of depolarization. Again, that changing from a negative inside to a positive as sodium's coming in. So this causes what's referred to as an action potential. And that follows kind of an all or none law. So it takes a certain threshold. So, just 1 or 2 sodiums coming in, that won't generate an action potential. But once it reaches a threshold, then this action potential gets generated that can flow down the axon. There's also what's referred to as a refractory period, and this is when those potassium voltage gated channels open. After a little delay, they're a little slower than the sodium, but once they do, now the potassium goes out of the cell. And that's why we need those sodium potassium pumps to put everything back the way it's supposed to be. Because during a nerve impulse, sodium comes in, potassium goes out, which is the opposite of what we started on. Again, as that potassium moves out, now we're making the interior more negative, and that causes those voltage gated sodium channels to become inactive or kind of closed. So now, during this kind of period after the action potential is passed, but before we get back to the resting, that's referred to as the refractory period. So again, it's that period between when the action potential was generated and when we're back to rest. All right, and so what we see here is one of the key biological processes in understanding this nerve impulse. There are multiple videos that are posted for you in eCampus, or you can find your own on YouTube or other sources so that you can really visualize what is happening. So I highly encourage you to look at those for this nerve impulse. But as the figure here is showing, and we'll continue on the next slide. So again, we have these voltage gated channels that are here. So we have the sodium in purple and the potassium in green. So at rest, again, the sodium is shut. Sodium can't come in. Potassium can kind of move through its channels. But for the most part, again, sodium is high outside, potassium is high inside. And again, we get this membrane potential. So the outside is more positive, the inside of the cell is more negative. Again, here's our sodium-potassium pump that's keeping everything the way it's supposed to be, where sodium goes out and potassium comes in. So this is here at rest. The inside is negative, the outside is positive. Our sodium-potassium pump is putting everything in order. All right, when there's some type of event, so again, touching something hot or some type of activity, and there's an influx here of sodium into the cell. Now, as sodium comes in, what it's showing over here is now the inside is more positive, the outside is more negative, because again, sodium is positively charged. So as it rushes in, the inside of that cell becomes more positive, the outside becomes negative, and so this is that depolarization. It's opposite to what it is at rest. Okay, so that's a depolarization. As sodium comes in, it changes or flip-flops. Now the inside is positive, the outside is negative. Now what that does is it's going to, as that sodium comes in and this becomes more positive, now it's going to open more voltage-gated sodium channels. So now more sodium comes in, and now further down this axon, we get this depolarization event where the inside becomes positive and the outside becomes negative. And then there's also a point to where these potassium, voltage-gated potassium channels open. Again, they're a little slower though, but as the potassium is coming out of the cell, that's going to lead back to this kind of repolarization. Let me erase some of this. All right, so during this refractory period, now we're going back to normal, to where the inside is more negative, the outside is more positive. But it's shown here in green because actually during this refractory period, even if there's like another trigger, those sodium channels cannot be activated. So there cannot be an action potential during this refractory period while we're waiting for everything to get back to normal. All right, so now what happens as that action potential, as that nerve impulse jumps from one node to the next down the axon, what's going to happen is once it reaches the end, now that sodium can't cross into the next neuron. So we have what's called a synapse. And so axons, so neurons don't physically touch one another, so they can't like pass on their ions or anything. There's a little gap. It's about 10 to 20 nanometers, not something you need to know for a test or anything, but just to give you an idea of how small we're talking. There's a small little separation between one neuron and the next called a synaptic cleft. And this junction of one axon to another is referred to as the synapse. So just some terminology that goes along with that. So the neuron before the synapse is called the presynaptic membrane, and then the neuron it's communicating with is called the postsynaptic. So it's receiving the information. The presynaptic is sending the information. So if we look here at the synapse with kind of this 3D modeling here. So here's our skeletal muscle. That's where the effector is going to be. Again, we have this mitochondria in here. But here we have an axon terminal. And so what it's going to do, you'll see all these little dots, these little pink dots that are here. These are called synaptic vesicles. They have neurotransmitters in them. And as that action potential comes down, it causes a release of those synaptic vesicles where they release neurons. So this is our presynaptic neuron. It has these vesicles with neurotransmitters. Then we have this gap that separates it from the postsynaptic. In this case, it's actually a muscle, or it could be another neuron, like an interneuron or just another neuron it's communicating with. So this is receiving the signal, receiving that neurotransmitter from the presynaptic. All right, now these neurotransmitters that get released, again, they're just a chemical messenger. So this is how we convert the electrical, the action potential, changing the positive and negative charges across the membrane. When it gets to the terminal, when it gets to the end, that electric signal can't cross the synapse. So it gets converted to these chemical messengers. and it releases neurotransmitters. So they're packaged, as we had just seen in the picture, in these tiny little vesicles called synaptic vesicles. So when that nerve impulse, that action potential, reaches the end of the axon, the vesicles release the neurotransmitters into that synaptic cleft. Now, these are chemically gated channels. They're going to bind to the postsynaptic. So these neurotransmitters bind to the postsynaptic membrane. Now, what they're going to do is whenever they bind, it's going to open a channel on the postsynaptic cell. All right, so this is just kind of visualizing. And again, there are some really cool videos in eCampus I encourage you to watch. But as that action potential is kind of jumping down these nodes of Ranvier, whenever we get down here, that influx of sodium tells the synaptic vesicle here to fuse with the membrane, and it releases its little neurotransmitters. And those neurotransmitters bind to ligand-gated or chemically gated channels. And whenever they bind, as this next picture is showing, whenever that chemical or ligand binds, now that ion is able to open. So it's closed, but once the ligand or chemical binds, then it opens up that gate and it allows that that ion to move through it. All right, so then what do those neurons do? Or what is the point? So there are actually excitatory and inhibitory synapses, meaning the signal being released can excite the next cell or it can inhibit it. So there are lots of different neurotransmitters. We are not focusing on all of them, but they essentially fall into two classes. They're excitatory or they are inhibitory. They both bind to an ion channel, so a chemically gated or ligand gated channel. The excitatory ones are going to usually be sodium channels. So when the ligand binds or the chemical binds, it opens them up, sodium rushes into the cell, and that leads to an action potential. That's why it's called excitatory. Now, an inhibitory synapse, it still releases a ligand, but when that ligand binds to a channel, it is something like potassium or chlorine, and opening of those channels are going to further make the inside more negative, and those will prevent an action potential. So they're both causing ions to move through the membrane. The difference is that an excitatory leads to an action potential and inhibitory does not. An individual nerve cell can actually have both excitatory and inhibitory synapses. All right, so it doesn't have to just be one or the other. Some of them couldn't be either one. So then how does the neuron know which one to release? Well, that's the job of what's called integration. So that has to happen actually before the signal is sent down the axon. So the region right at the beginning of an axon is called an axon hillock. And so there, this integration occurs where it's going to take in all the information, so the various excitatory and inhibitory electrical effects. It is going to interpret what actually needs to happen. Do I need to generate an action potential or do I need to stop? So that type of taking all the information in is done at the axon hillock. So if the excitatory wins out, it sends the action potential down the axon. If not, then it doesn't. So again, there's a threshold. So that depolarization, that change in the resting membrane potential has to be big enough that an action potential actually gets fired. This is just showing that integration. So again, at one cell body, it can have multiple signals coming in at one time. So some that are inhibiting it, some that are activating it, others, again, lots of different signals coming in, kind of overload for this cell body. Okay, but here at that axon hillock, right at the beginning of the axon, this is where that integration happens. It takes into account everything happening to this neuron, and it determines, is that enough change in the membrane potential to start an action potential and cause the release of neurotransmitters, or it's not enough of a change, everything's kind of canceling out, and it just remains at rest. All right, so a few different types of neurotransmitters. There are many, many, many. We're not covering them all, but a few of the key ones. So acetylcholine, this is one of the excitatory neurotransmitters. It's typically found at muscle fibers, so it's going to cause contraction or activation of a muscle. So where the neuron and the muscle fiber come together is called a neuromuscular junction. And again, this is an excitatory synapse. So when acetylcholine gets released from the neuron onto the muscle, it causes skeletal muscle to become excited. But at the same time, even though it's the same neurotransmitter, it actually inhibits cardiac muscle. So the same neurotransmitter can have different effects depending on the receptor that it binds to or the ion channel it binds to. So again, it excites skeletal muscle, it inhibits cardiac muscle. Glycine and GABA are the two main inhibitory neurotransmitters. So they're going to make it harder for an action potential to be generated. So it's going to make the inside more negative, making that action potential harder to reach threshold. Just an example here, Valium enhances the binding of GABA. So that inhibits the brain, it kind of slows it down. So it's a relaxant. Some others, so these have biogenic amines, and there are a few of these that are pretty common to us. So dopamine, norepinephrine, and epinephrine, and then serotonin. So dopamine has a lot of jobs in the brain. It controls body movement, but it's also really important in our reward system. Whereas norepinephrine and epinephrine, this is kind of like our speed, it activates our autonomic nervous system and causes kind of that fight or flight response. And then serotonin is involved in sleep, but also our emotional state. So our feeling of happiness is regulated by serotonin. So the drug PCP actually blocks the elimination of these amines. And it produces a lot of different effects. So if it blocks the elimination from the synapse, that means they're staying there longer. So it enhances their action. All right, we also have what are called neuromodulators. So again, the body sometimes deliberately prolongs the transmission of a signal by slowing the destruction of the neurotransmitters. So those are called neuromodulators. So there are special long-lasting chemicals that aid in the release of neurotransmitters and or delay the breakdown of neurotransmitters. So either way, whether it's causing the release of them or it's stopping the breakdown once they get in the synapse, it's causing that signal to stay in the synapse longer to prolong the signal that's there. All right, whenever we talk especially about dopamine, we need to talk a little bit about addiction. So addictive drugs act on these synapses. So again, our emotional states, our mood and pleasure are determined by specific neurotransmitters and neurons, particularly dopamine and serotonin. So we now are pretty confident that depression results from a decrease in serotonin. So medicines like Prozac, that's an antidepressant, it inhibits the reabsorption of serotonin. So it modulates serotonin, keeps it in the synapse longer to make an individual feel happier than they would without the medication. All right, so again, just kind of looking at this alteration here. So again, our presynaptic cell here, it's releasing all these neurotransmitters. Typically, the neurotransmitter is there just for a short amount of time. Some of it gets taken back up into the presynaptic cell. That's called reuptake. Of course, some of it binds to receptors. Some of it just diffuses away. And other times there are like enzymes here that break down or chop up that neurotransmitter to get rid of it, because typically we don't want it to stay very long unless we keep having a signal. So again, if you touch something hot, your reflex removes your hand. As long as you're still not touching the hot plate, you don't need to keep sending that signal, so you can stop it. However, these modulators, what Prozac does is it blocks the reabsorption. So now the serotonin does not go back into the cell, and it leaves it to hang out a little bit longer in the synapse, and then eventually it reaches that receptor to cause an effect. So it keeps the serotonin around longer. Right. Sometimes, though, there's a loss of sensitivity. So the nerve cells essentially kind of ignore, kind of like a mom whose kid keeps going, mom, 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 and you just kind of tune it out. Well, nerves get like that too. If it keeps getting bombarded by a neurotransmitter, it's kind of like, I'm done with you. I'm not going to listen to you anymore. I'm not responding to you anymore. So whenever this happens, when that signal's exposed for a long time, then those receptors that it's binding to, if it's there too long, they often insert fewer receptors. And this is what can lead then to addiction. So for example, cocaine is a neuromodulator. So it causes abnormal, like crazy amounts of neurotransmitters, especially dopamine, to remain in the synapse for a long time. And this affects the pleasure pathway, so it makes the individual feel happy because of the release of dopamine, but it also is a reward system, so it remembers that it felt really good whenever it took this compound. Again, that's going to lead to that addictive behavior. But what the cocaine does is it blocks the transporter protein on the presynaptic, a lot like Prozac. So now the dopamine does not go back to the presynaptic cell. It doesn't get reabsorbed, so it stays there longer. So because it can't bind to that transporter to go back to the presynaptic cell, it stays in the synapse longer and continues to stimulate it. And again, this is what ultimately leads to addiction. So when those proteins in the pleasure pathway are exposed to high levels of dopamine, which absolutely cocaine releases huge amounts, as well as most addictive drugs, ultimately they release large amounts of dopamine. And so again, the cells are like, if you're going to give me this so much dopamine, I'm going to stop paying attention to you. So they lower the number of dopamine receptors. Well, now, because there are so few receptors, now the drug user needs more of the drug to maintain normal nerve activity, because again, it doesn't have as many receptors. It's adapting to that large signal. So it produces less receptors. Now you need more, a higher dose than before. This leads to tolerance. Now we need more of the drug to get the same effect. That further feeds in that reward system and ultimately leads to addiction, which is the physiological adaptation due to drug abuse. Now the body feels like it has to have it in order to be at normal, because we've reduced those receptors, we've changed the chemistry in the brain, and now we need that drug to feel normal. So again, this is just kind of summarizing in a picture kind of form. So again, here's our dopamine receptor. So normally, again, neurotransmitter comes out, some of it binds to the receptor, but some of it binds to this transporter and goes back into the presynaptic cell, and that's called reabsorption. Okay. However, if we have a drug like dopamine that blocks that reabsorption, like Prozac, now this dopamine stays longer in the synapse, so more of it is going to bind to all these receptors. Well, if the receptors keep hanging out, then eventually what happens is it's just going to remove some of these receptors and break them down. So now after continued use, now we have fewer receptors. So now whenever that drug's removed and dopamine is released, there are fewer receptors to receive it, and most of it ends up going back in to the presynaptic cell, and no effect or little effect is seen on that postsynaptic. So now more of the drug needs to be released to get those normal levels to activate these receptors. All right. And then nicotine, just another one, of course, with vaping, with cigarettes, chewing tobacco, whatever the case may be, anything with nicotine, it is actually one of the most addictive drugs. And what it does is it binds to the postsynaptic receptors in the brain that normally bind to that acetylcholine. And so whenever it does that, now the brain responds. Again, it adapts. So it produces fewer receptors that nicotine can bind to, and that alters the pattern of activation of those nicotine receptors. Okay, so normally acetylcholine would bind to them, but now because there's so much nicotine, the body's like, you know what, I'm done. I'm going to stop making these receptors. So now those normal responses, when acetylcholine does get released, there are fewer receptors there to do that job. So prolonged effect or prolonged changes in those patterns ultimately lead to the addictive nature of nicotine, because more and more of it has to be achieved because we have fewer receptors to be activated once we prolong the activity or the use of that drug. So again, nicotine is again one of the most addicting drugs or compounds that are out there. Not only does it affect the acetylcholine, but it also releases dopamine that feeds into that pleasure center.